Welcome everyone. Today we are going to learn about phosphoglycerides in more details. In my previous video, I have discussed about the lipid components of plasma membrane, and in that video I have mentioned that there are three major categories of lipids present. One is phosphoglycerides, another is sphingolipids, and third is cholesterol. Now in this video we will learn about phosphoglycerides in more details. So today's topic is phosphoglycerides. Phosphoglycerides. Now the name suggests here that there must be a phosphates and glycerols. I can write the major components of the phosphoglycerides. The first component of phosphoglycerides is glycerol, as the name suggests, glycerol. And one thing we have to remember that glycerol is the backbone in this molecule. So glycerol is the backbone of all phosphoglycerides. The second components of the phosphoglycerides is two fatty acid chains. So there must be two fatty acid chains. The third component is that a phosphate group should be there. Phosphate group. So that's why it is called phosphoglycerides. Phosphate group there must be. The fourth component is alcohol head group. Okay, now let's talk about each component in more details. As I have mentioned that glycerol is the backbone. Glycerol is actually a 3-carbon molecule. So let me write glycerol basically has 3 carbon molecule. Let's suppose this is the first carbon, this is the second carbon, and this is the third carbon. With the first carbon there is a fatty acids actually attached. With the second carbon another fatty acid is attached. So there is 2 fatty acids attached with the glycerol backbone. Now with the third carbon a phosphate group is attached. A phosphate group is actually attached with the third carbon. And with the third carbon, with the phosphate group and a head group is actually attached. So alcohol head group is actually attached. Now togetherly this portion, actually this alcohol head groups and the phosphates, is the hydrophilic portion of this molecule. Okay, this is the hydrophilic portions of the molecule, and these 2 fatty acids are actually hydrophobic in nature. Hydrophobic, that means water fearing, and this is the water loving portions. Hydrophobic. Okay. Now as these phosphoglycerides contain both the hydrophilic and hydrophobic portion, we togetherly call this type of molecule amphipathic molecule. Amphipathic or amphiphilic molecules. You can remember any of them. Okay, so this is the basic structure of phosphoglycerides. Now I want to mention that the bonds between the first carbon and fatty acids, the bond between the second carbon and the fatty acids, and the bond with the third carbon and phosphates, all are ester in nature. Remember this, all bonds are ester in nature. So this is the information about glycerol. Now let's talk about fatty acids. Okay, commonly it has been found that the length of these fatty acids, commonly the length of the fatty acids in the plasma membrane is 16 or 18. The number of carbon in the fatty acids is commonly 16 or 18, but sometimes carbon 14 is present. Carbon 14 fatty acids, those fatty acids contain carbon number 14, we call it myristic acid. And with 20, carbon number of 20, also there are some fatty acids also present. This C20 fatty acid are called arachidonic acid. So basically, commonly there are C16 and C18. I can write here C18 fatty acids are actually present. Now, one information I want to share about the fatty acids, that a fatty acid may have a double bond, and the formula about the fatty acids, formula of the fatty acids is actually C X is to Y. Now the X is equals to the number of carbon in the fatty acid, like 16, 18, 14 or 20. This is the number of carbon, and Y is the number of CC double bonds. So as I have mentioning that fatty acids may contain double bonds, so Y represent the number of double bonds. Now according to the presence or absence of double bonds, we actually discriminate the fatty acids. If the fatty acid don't have any double bonds, we call the fatty acid saturated fatty acids. So saturated fatty acids means there is no CC, sorry, it is the number, it is no CC double bonds. So there will be no CC or carbon-carbon double bond. This is a saturated fatty acid. And we call unsaturated fatty acids those fatty acids contain at least one or more greater than one carbon-carbon double bond. At least one or more than one carbon-carbon double bond. Now, if the unsaturated fatty acids contain single, let's suppose one carbon-carbon double bond, we call it mono unsaturated fatty acids, or in short we can call it MUFA. And if the unsaturated fatty acid contain more than one, so this fatty acid contain a single CC double bond. And if the unsaturated fatty acid contain more than one carbon-carbon double bond, we call it poly unsaturated fatty acid, or in short PUFA. Okay, here greater than one carbon-carbon double bond is present. So according to the presence of double bond, we can discriminate the fatty acid into saturated, unsaturated, and also mono and poly unsaturated fatty acids. So now one information I want to share, that if any fatty acid, let's suppose here, if any fatty acids are saturated, then the fatty acid is actually straight. The shape of the fatty acid should be straight. But if there is any double bond is present in the fatty acid, then there is a kink or bend in the structure. Let's suppose here is a CC double bond, then the fatty acids will look like this. The fatty acid will come down here, but it will actually become like this. So there must be a kink. So where there is a double bond, there will be a kink in the structure. Okay, so if there is more than one double bond, then there will be another kink. Let's suppose if there is another double bond here, then the structure of the fatty acid will be like this. So, and this has an important function in the plasma membrane. I will talk about in another class. So where there is a cis double bond, so this double bond is actually cis in nature. Where there is a cis double bond, then the bond actually makes the bending in the structure is called kink. So this is all about the fatty acid chains. Now phosphate group is also present. Now another important point we have to remember, that is the alcohol head groups. The alcohol head groups could be choline, and if this is a choline, we call it phosphatidyl choline. Phosphatidyl choline, or in short we call it PC. Then another head group could be ethanolamine, so we will call it phosphatidyl ethanolamine, or in short we call it PE. If the head group, alcohol head group is serine, we can call it phosphatidyl. The name is same, phosphatidyl serine. And if the head group is inositol, then we will call it phosphatidyl inositol. Now, at physiologic pH means approximate 7.2 physiologic pH. So at physiologic pH, the pH that is present in the blood or in our cell, physiologic pH, the phosphatidylcholine and phosphatidylethionamine has no net charge, so they are electrically neutral, so they don't have any charge, phosphatidylcholine and phosphatidylethionamine. But serine and phosphatidylinositol, this phosphatidylserine and phosphatidylinositol at the physiologic pH, at pH 7.2, they have a single net negative charge. This has also important role. Now, one thing I want to mention, that in general it has been found that the fatty acids attached with the carbon 1 has no or a single double bond, but the fatty acid that is attached with the second carbon of the glycerol, they have one or more than one double bond.